All right, Nerds, in this video we're going to talk about glycogenesis. So the question is, what is glycogenesis? Well, if you look at the name or the word itself, it's saying glycogen synthesis. That's all glycogenesis is. It's just basically glycogen synthesis. But the question is at hand is, what is actually helping to synthesize this glycogen? Okay, that's where we got to start. Okay. So generally, we're going to have two cells that are going to be playing a big role in this glycogen metabolism. There's many different types of tissue cells, but we're going to focus on the significant ones. What are those two main organs that are going to be playing a role in this glycogen metabolism? One of the organs is going to be the liver. This is going to play a very crucial role in this actual glycogen metabolism. Now, on average, with this glycogen metabolism, about how much glycogen can be stored or generated within the liver. Because only certain amounts can actually be stored. We can't go beyond a certain capacity, and that is about 100 grams approximately. So about 100 grams of glycogen can be stored in the liver. Conversely, let's say that we have the muscle. So the muscles are also going to play a very important role with this glycogen processing. Specifically, they can actually store up to about 300 to 400 grams of carbohydrates in the form of glycogen. So 300 to 400 grams of glycogen can be stored in this organ here. So the liver and the muscles are playing a crucial role in this glycogen metabolism process. Specifically, we're going to focus on glycogenesis in this video, and we'll have another video where we focus on glycogenolysis, which is just the reverse pathway. The next question at hand is, when is this glycogenesis process going to be occurring? So when does it occur? So we know the organs that it's occurring in, but when is it occurring is the question. So when is it occurring? It's occurring whenever you are feeding, right? So whenever you're actually eating food, you're ingesting food. So we call that, it's occurring during the fed state. The fed state is basically the absorptive state. It's whenever you're eating food and you're absorbing that actual glucose molecules across the GI tract and into the bloodstream. That's the fed state. So generally, what will we say during the fed state? We would say that this person has high blood glucose levels. What do you call that when your glucose levels are really high? We call that hyperglycemia. But we're gonna, this is not pathological hyperglycemia. Normally the range of glucose is about 80 to 120 milligrams per DL, give or take 10 below, 10 up. So you can technically say, if you want to give it a decent range, about 7 to 130. Whenever you're going significantly beyond 130, it can become pathological. But if you're just eating a decent amount of food, it'll go up a little bit, right? Enough to trigger the production of a very special hormone that we're going to talk about afterwards called insulin. Insulin is a hormone produced by the pancreatic beta cells. And it's going to help in the regulation of this process. Okay, so we know the organs that it's occurring in. It's occurring in the liver, it's occurring in the muscles. We know when it's occurring. It's occurring in the fed state whenever our blood glucose levels are really high, hyperglycemia, so we've just eaten. And we also know the hormone that's helping to regulate this process, which is that of insulin. Okay, now what we got to do is exactly how it's happening. So now we have to actually answer the question, how? And that's what we're going to be focusing on primarily throughout this video. Okay, so the first thing that we have to do is we have to actually say, okay, what are we actually dealing with here? We're bringing the glucose into the cells. So then we have to take a look at glucose. So real quickly, let me draw for you the structure of glucose really quickly. So let's say here, I draw an expanded version of the glucose molecule. So it's a 6-carbon glucose molecule, and usually it's in a cyclic form. Okay? So it looks something like this, right? And usually there's actually going to be a carbon group over here, and usually there's going to be an OH here and a hydrogen here, and then sometimes there could even be a hydrogen and an OH over here, and vice versa. We could just keep going all the way around. What's the significant component of this, though? Over here also in the 4-carbon there can be a OH. Let me number the carbons for you. So this is the general structure of our glucose molecule. Now what I'm going to do is I'm going to number the carbons, because the number of the carbon is going to be the specific point that we want to understand. So let's say right here, this one right there that's right next to the oxygen, this is actually the number 1 carbon. Okay? That's the number 1 carbon. This is the number 2 carbon, number 3 carbon, number 4 carbon, 5 carbon, and 6 carbon. The reason why I'm telling you this is that we're going to talk about certain bonds that are going to be occurring between the 1 carbon and the 4 carbon. I'm going to briefly introduce it because we're going to talk about it throughout the process of this video. For example, you know the number 1 carbon, we refer to the number 1 carbon as the reducing end. So we refer to it as the reducing end. And the reason why it's called the reducing end is that it actually has a carbonyl carbon. So you know carbonyl carbons are specifically looking something like this. In general, they have carbonyl carbon. That's the general structure of a carbonyl. That number 1 carbon is the reducing end. It's a carbonyl carbon. At the number 4 carbon, we have this specific hydroxyl group. So let me say for a second, I take this glucose molecule with its number 1 carbon. And what I'm going to do is, I'm going to take another glucose molecule and I'm going to link it up to the 4 carbon. So let me say I take over here, I draw another 4 carbon, I draw another 6 carbon glucose here. So let's say here I have another 6 carbon glucose. And then here should be a oxygen. Here should be our CH2OH. Here should be the OH, hydrogen, and what did I say over here? Specifically, you should have a OH and even a hydrogen right there. And again, what is this carbon here? This is 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, 6 carbon. What I'm going to do now is I'm going to show you a special bond that we're going to be talking about. So I don't want you guys to just be confused when I just throw that out there. I want you to understand what's exactly happening. You see this number 1 carbon and this 4 carbon? The 1 carbon I'm going to denote again as our reducing end. I'm going to denote that with R. The number 4 end is going to be the non-reducing end. What I'm going to do is I'm going to take this 1 carbon and this 4 carbon and I'm going to fuse them together between these OH groups. So these OH groups are actually going to fuse together and you're going to lose water out of this process. So you'll actually lose water. So out of this will come water. But then look what happens now. I'm going to draw a new bond here between these 2 guys here. I'm going to actually draw a different oxygen. So here's my oxygen right there. And then you're having a bond here and a bond there. So you see this bond that I just made between this 1 carbon and that 4 carbon? That's a special type of bond that we're going to talk about. That bond right there is actually called a alpha. And the reason why you know what they call it an alpha is, for example, if I had an OH pointing up this way and an OH pointing up this way, and I form the glycosidic bond between them when they're both upwards, that's alpha. If it was opposite, let's say that I had an OH down here and I had an OH pointing up here. So one's pointing down, one's pointing up. And I form the glycosidic bond like this. That's beta. So this is a beta and this is alpha. Specifically, what type of bond is this going to be? Well, the OHs were both pointing up. So this is an alpha 1,4 glycosidic bond. Okay, so now we understand what the alpha 1,4 glycosidic bond is. It's between the 1 carbon and the 4 carbon, and it's just going to be the formation of a hemiacetal. Okay, we'll talk about that in organic chemistry with this OH of the 4th carbon. Okay, with aldol condensation reactions. We'll talk about that in orgo. Okay, now I need to show you one more bond, and then we're going to get into this process. And that's why I mentioned this 6 carbon, because I highlighted a whole bunch of them, but specifically this one's another important one. So let's say I take this 6 carbon, and I take another glucose molecule, and I link it up here. So now I draw another glucose molecule. Let's say I draw one like this. Here's my oxygen right there. Here's my CH2OH. That's my 6 carbon again. And this is my 1 carbon. And again, what's coming off of the 1 carbon? It's going to have a OH and a H here. And then over here, I'm going to have a OH and an H. And just again, just to denote our carbons, what is this carbon here? This is specifically 1 carbon. This is 2, 3, 4, 5, 6. Now what I'm going to do is I'm going to take this OH from the 6 carbon and I'm going to fuse that with the OH of the 1 carbon. I'm going to have a hemiacetal to an alcohol reaction. So I'm going to get rid of this. I'm going to get rid of this. I'm going to release water out of it. And then what's the bond that I'm actually going to form here? I'm going to form a bond between the 1 carbon of this glucose molecule and the 6 carbon of this other glucose molecule. That bond is, again, pretend that the OHs are both pointing in the same direction. So what kind of bond would this one be? This would be a alpha 1,6 glycosidic bond. Now that we have an understanding of what these are going to look like, so that way when I go through that process, it makes sense. I'm not going to draw every single glucose molecule like this. We're going to try to make it easier. So here's what I want you guys to imagine for the rest of this process. Here's a circle. That's our glucose molecule. And then I want you guys to just realize that if I have this glucose molecule, let's say that one end is going to be a 1 end, then I could have a 4 end, and let's say that I have the arm coming out of it, which is this going to be the CH2OH end. What would that be? This would be the 6 carbon. And to give you a different color so that way it's completely clear here, again, one end over here could be the 1 end, one over here could be the 4 end, and this could be the 6 end. And if you want for me to even make it simpler, I could even put a smiley face in here so that the eyes are actually going to be towards the top. Okay? So now what we're going to do is we're going to show the bonds like this. We're going to show glucose like this because it's easier, it's simpler. Okay, so now what we're going to do is we're going to go through this process. Okay, so now that we've gone through that part of it, just the general structure, let's make it simpler on ourselves and let's go ahead and get this started. So now, like I told you, I'm going to represent it as a circle. And if you guys like, I'll put a little smiley face in there so that you guys can think this is a happy process. Let's have fun throughout this process, guys. So again, there's his little point there. And again, if we were to number it just for the sake of it for the beginning parts, 1, 4, 6. Okay, this is our glucose molecule. Now, in order to get this glucose molecule into this liver cell, let's actually focus specific on a liver cell. Let's do liver cell first. And the same concept applies to the muscle cell, just a little bit different in regulation and enzymes. If I bring this glucose into the cell, I have to have a special type of transporter to get that puppy in. If it's the liver, it's a GLUT2. So if this is the liver, this is a GLUT2 receptor. If it was the muscles, it's a GLUT4. Okay? But now what's going to happen as a result of this is this glucose molecule is going to get brought into this cell. So now the glucose molecule has been brought into this actual liver cell. As it's brought into this liver cell, it's going to be acted on by a special enzyme. What is that enzyme that's going to work in this step right there? This enzyme right here is ready to help. If it's in the actual liver, it's special. This enzyme is actually called glucokinase. So glucokinase is stimulating this first step here. But remember, glucokinase is specifically for in the liver. If it was in the muscles, it would be hexokinase. Okay? So if it was in the muscles, here, we'll put that just for the sake of it, up here, hexokinase would be specific for the muscles. Now, what is this hexokinase or this glucokinase going to do? They're going to take a specific molecule called ATP. And they're going to react this into this reaction. They're going to put ATP into the reaction. When they put ATP in the reaction, look what ADP is going to come. It's going to come out as ADP. That means that in this process, it lost a phosphate. You know what happened to that phosphate in this reaction? We put the phosphate on the 6 carbon of this little glucose baby. So now let's actually show the result here. So here's our glucose baby. And here again, that's our 6 carbon. But what's going to be on the 6 carbon? Let's show it as like a little dot here. What's this little dot? That's the phosphate, right? So this little dot here that we added on is going to be the phosphate. Then if you remember, again, if we have his little eyes here, smiley face, this would be the number 1 carbon, 4 carbon, 6 carbon. What is this molecule now called? This molecule is now called glucose 6-phosphate. Okay, because the phosphate that we added on is on the 6 carbon. But you know what? This glycogen metabolism doesn't want glucose 6-phosphate. We want a specific type of glucose. We want it to be preferred only for glycogen metabolism. So you know what happens? Our body has a special enzyme, and this enzyme is reversible. It can do both reactions. It can convert it up and down. And what it does is it just shifts the phosphate from the 6 carbon to the 1 carbon. So now what would this glucose molecule look like? Let's say here it is. And again, what's going to be here? The smiley face, right? And then over here you're going to have, again, the 6 carbon is going to be popping out like this. That's our 6 carbon. And then what are we going to have now? Specifically, 1 carbon, 6 carbon, 4th carbon. And on the 1 carbon, we are now going to have a phosphate group. So there's our phosphate group. And it's specifically on the 1 carbon. If the phosphate group is on the 1 carbon, what will we call that? We're going to call this glucose 1-phosphate. What enzyme is catalyzing this step? The enzyme catalyzing this conversion, this downward direction, but again, it can do both steps, up and down, is called phosphoglucokinase. mutase. So this enzyme is not extremely regulated, and that's why it's reversible, and it's called phosphoglucomutase, and it's a reversible enzyme. It takes glucose 6-phosphate to glucose 1-phosphate. Now we have the basic precursor, but glycogen metabolism is still like, I want it to be a little bit different. So then they see something. They're like, okay, what I really want is I want you to take this glucose 1-phosphate and I want you to combine it with a special molecule. So there's a molecule here which is called uridine triphosphate. So uridine triphosphate is basically, if I were to kind of just give you the basic structure, you know you have what's called nucleotides, and nucleotides, a specific one is called uracil. That uracil is a nitrogenous base, and it's bound to what's called a ribose sugar. That ribose is then bound to 1, 2, 3 phosphates. Uracil and ribose is called uridine. And then with the 3 phosphates, it's triphosphate. So it's uridine triphosphate. This uridine triphosphate is going to combine with the glucose 1-phosphate. And what's going to happen is this carbon on this, the 1 carbon with the phosphate, and then this UTP, you're going to break this bond right here. So look, I'm going to break this bond. This bond is going to be broken. And what do I get out of this reaction? What am I going to lose as a process of this reaction? I'm going to lose 2 phosphates. These 2 phosphates that I lose, that are bound together, has a special name. This special name is called pyrophosphate. Okay, then I have the uracil and the ribose, so the uridine monophosphate is going to combine with the glucose 1-phosphate. And as a result, what am I going to get? I'm going to get a 1-phosphate, another phosphate. Oh, that's diphosphate. So now it's going to be so it's going to be called uridine diphosphate with a glucose link to it. So we call it UDP glucose. So as a result of this reaction, of us fusing this UTP and this glucose 1-phosphate, what am I going to get as a result of this reaction? I'm going to get specifically UDP, which is your uridine diphosphate, which is the 1-phosphate here plus the phosphate from the 1 carbon of glucose. And what am I going to have linked to it? Right here. I'm going to have, there's my 6 carbon, smiley face right there. This is my glucose. So this is called UDP, and let's mark this guy. This is glucose. And for the sake of carbon numbering, this is 1 carbon, 6 carbon, 4 carbon, right? Now we have the basic unit of what we need to make the glycogen. But the problem is, in order to make glycogen, it costs energy. It's an energy costing process, right? It's an endergonic process. In other words, the delta G of this reaction is positive. Our body has a weird way of coupling reactions. We can chemically couple reactions. So what we have over here is this pyrophosphate. I'm going to break this bond because it's going to release a lot of energy. And there's an enzyme that's catalyzing this process here. This enzyme is called pyrophosphatase. And what he's doing is he's stimulating the breakdown of this pyrophosphate. And what am I going to release as a result? 2 different phosphates, an individual inorganic phosphate here and an individual inorganic phosphate here. But as a result, what's going to happen? This is going to produce energy. This is going to give off energy. And this is a negative delta G. The delta G is negative for this reaction. If the delta G for this reaction is negative, that's an exergonic reaction, which can drive this endergonic reaction. It's chemical coupling there. Okay, this UDP glucose, what's going to happen here? I'm going to have a base molecule that I'm going to have to build on. Let me put this base molecule here. Here's this base molecule. This base molecule that I'm going to have here is already going to be present within this actual liver or this muscle cell. And it's a special protein molecule called glycogenin. And what's special about glycogenin is he has specific amino acids that are coming off of his surface. What are those amino acids that's coming off of its surface? Especially tyrosine. So tyrosine, you know, it has a phenyl group, and then off that phenyl group you're going to have an alcohol. So this is going to have that alcohol poking out. That's attractive to this UDP glucose. So what has to happen is now what I'm going to do is I'm going to take this UDP glucose And I'm going to take this UDP glucose, I'm going to add it onto this glycogenin. So again, what is this brown protein molecule here? It's called glycogenin. What I'm going to do is, and I'm going to take this glycogenin, for the basic units of the glycogenin, I'm going to take this UDP glucose and I'm going to add it on. So let me add this on now. The interesting thing is that some of these UDP glucose molecules that I'm going to add on, it doesn't require a specific enzyme. In other words, this process is what's called autoglycosylation. Glycosylation. So autoglycosylation means that there's not going to be a specific enzyme in this process that's going to add these UDP glucoses on. So now let's add a couple UDP glucoses onto this glycogenin molecule. So I'm going to make my glycogenin a little bit smaller here. And again, what am I going to have linking them? Well, first off, I'm going to have a OH. But let me just get rid of that H, because now what I'm going to do is I'm going to link this glucose on the one carbon. So now what I'm going to do is there's a phosphate right here, right? There was a phosphate that was having the uracil diphosphate with the glucose. Now what I'm going to do is when I add this UDP glucose on, I'm going to break this bond right there. So I'm going to break this bond. And what am I going to do as a result? What am I going to release out of this process? I'm going to release out UDP. And that'll go on later to actually combine with the phosphate to make UTP. But then I'm going to combine onto this a glucose molecule. Okay, and then what would be coming off here to the side? This would be the 6 carbon. So here's my 1 carbon, 4 carbon, 6 carbon. But then guess what's going to happen? I'm going to do another one. So then I'm going to release another UDP. So say I do it again a second time. So this is the first time, this is the second time. And let's say I do it another time, a third time. Let's say I do it 3 times to get it all started. And I release out UDP, UDP. What is this going to look like as a result? Okay, well I'm going to take this next glucose and I'm going to add the 1 carbon onto this. Didn't I tell you that whenever we have a bond between the 1 carbon and the 4 carbon, that's something really special? Darn right it is. It's called a alpha-1,4 glycosidic bond. So now I'm going to take this next glucose molecule, the little smiley face, and I just link these two together. What is that bond right there? That is an alpha-1,4. Well, this is the first one. This is going to be the second one, and then I'm going to do a third one. And what am I going to do here? I'm going to add another glucose molecule on here. So now this is going to be the 6 carbon and 6 carbon. So you see what we've done here. There's a couple autoglycosylations that are occurring before any enzyme is involved here yet. All I'm doing is I'm taking the UDP glucose, breaking the bond between the UDP and the glucose, between the 1 carbon of glucose, breaking that bond, releasing the UDP, and taking the glucose and adding it onto this. What is this brown molecule here called? This is called glycogenin, which has a special tyrosine amino acids around it. But this is, again, not requiring an enzyme, just a couple glucose molecules to prime it. This is not requiring an enzyme. Maybe we just do 3 for right now. And again, I add the UDP glucose. When I do that, I get rid of the UDP, add the glucose with the 1 carbon onto this glycogenin molecule. This is not a glycosidic bond. Okay, this is not a glycosidic bond because this is a bond between a glucose molecule and a protein. That's not a glycosidic bond. This bond between this glucose and this glucose, from the 4 carbon to the 1 carbon, that's an alpha-1,4 glycosidic bond. And this glucose molecule to this glucose molecule, link between the 4 carbon and the 1 carbon, is also a alpha-1,4 glycosidic bond. So again, this bond right here and this bond right here are specifically what? Alpha-1,4 glycosidic bonds. Okay, so now we have our primer. So now let's bring this primer over here and do the next step. So now what we're going to do is we're going to bring this guy over here. And we're going to continue to build upon him. So let's say here is my glycogenin molecule. And already I have how many glucose molecules linked to it? I have 3. So let's draw those 3 glucose molecules already previously linked to this one. So again, I'm going to have right here 1, then I'm going to have 2, then I'm going to have 3. And all of these guys are linked together specifically through, so far, 1,4 glycosidic bonds. So there's my 3 glucoses, and again, I got to draw 6 carbon, 6 carbon, 6 carbon. And if I were to be very specific, just to be consistent, this is 1, this is 4, 6, 6, 6. Okay? Now, an enzyme comes into play. This enzyme is very, very interesting. So let's draw this enzyme. Look at this dude, okay? He's going to do something really cool. He's going to help out in this process. What this enzyme is going to do is he's going to do something really, really special. He's going to continue to glycosylate this molecule. What do I mean by that? Okay. In one hand, he's going to hold on to this glycogen molecule. So let's say in one hand, he's holding on to this. The other hand, he has a UDP glucose. So what is that? Let's draw this over here. Over here in this other hand, he's going to have a UDP linked to a glucose. Okay? What's going to happen now is that he's going to take this UDP glucose and add it on and continue to do what was happening over here. If that happens, if I add this UDP glucose onto this guy, so let's say I keep adding a couple of these at a time. Let's say I add 4 of them. Let's add another one here, 2, 3, 4. So I added 4 of these. So now I have this guy, this guy, this guy, and this guy. And again, coming off of the side is going to be a 6-carbon molecule here. Right, we're going to have that 6-carbon. So again, here's our 6-carbon. And again, what did I do with this UDP glucose? I took the UDP glucose and I added on to this, right? So we had 3 previously. I just added on 4 more. When I add on these next 4, what am I actually forming? In between this third one, I form a alpha-1,4 glycosidic bond. In between this one, I form an alpha-1,4 glycosidic bond. Alpha-1,4, alpha-1,4 glycosidic bond. And as a process from this point here, this one, and this one, what do I release? UDP, 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 and UDP. All I'm doing is I'm elongating the glycogen. So let's say that I just keep doing that with this guy. This guy, this enzyme, he just keeps adding on more glucose molecules, one at a time, utilizing UDP glucose. And again, this is a 6-carbon, 6-carbon, 6-carbon. If he just continues to keep elongating this chain of glycogen, what is this enzyme called? And again, what would be happening as a result? What would I be releasing out of this? UDP, I'd be releasing UDP, and again, I'd be releasing UDP. This enzyme is just continuously doing that. He's just taking these UDP glucoses and adding them on consistently to each component. And if he continues to keep doing that, he's stimulating each one of these processes to continue to stimulate and make glycogen. What is the name of this enzyme? This enzyme is called glycogen synthase. Okay. So that's how he's doing that. Okay, cool. So far we had a nice polymer, but the problem is that, look, can we make this many inside of one cell? No. We got to do something special to where we can make it to where it's actually allowing for a nice surface area, but it's not clogging up the whole cell occupation of the cytosol. So how do I do that? I bring another enzyme in here. So now let us bring this polymer that we've made so far, let us bring him down here and see what happens to this next step, okay? Now, if we have this glycogen molecule here again, if I have this glycogen molecule here again, let's say here's our glycogen, and what is this glycogen having on its surface? Again, just repetition over repetition. It was having a tyrosine amino acid, and again, it was linking to a glucose, but this was not a glycosidic bond. And if we had, let's say that I do about maybe 10 of these. So let me just do a couple of these. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay. And all of these bonds in between that I'm going to shade in with a little bit of green here. This is a alpha-1,4, 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 alpha-1,4. Who is making this bond? Glycogen synthase. Another enzyme is going to come out. This enzyme is going to do something really cool. So again, what was coming off of the side over here, off of each one of these? 6, 6, 6, 6 carbon, 6 carbon, 6 carbon, 6 carbon, 6 carbon, 6 carbon. You guys get the point. What's going to happen is there's going to be a cool little, sneaky little enzyme who's going to do something really cool. So let's say here's this enzyme. This enzyme is going to come into play, and he's going to see all of this, and he's going to help out in this process. What is he going to do? In one hand, actually no, look, his foot. his foot is going to come down here and hold on to the glycogen molecule, this whole polymer. The other hand, let's say he counts down. Let's say he comes over here and he's like, okay, 1, 2, 3, 4, 5. I like that alpha-1,4 glycosidic bond. I'm going to cut that bond. So with this hand right here, he cuts that bond, okay? He cuts this alpha-1,4 glycosidic bond. When he cuts that bond, then look what he does. He takes with his other hand and transfers it onto the side. What bond is over here on the side again, guys? 6 carbon, 6 carbon, 6 carbon, 6 carbon. You guys get the point. It's the 6th carbon. This guy breaks that bond, and he takes 1, 2, 3, 4, 5, and transfers them over. Let's say he transfers them over to like this guy right here. This one right there. So now what I'm going to do is, I'm going to take these guys, 1, 2, 3, 4, 5. Each one of these guys, and again, this is the 1 end, and this is going to be the 4 end. And what I'm going to do, and this is the 6th end, let's say that's the 6th end, right there. I'm going to take, and now I got rid of these guys. He cut these and he transferred them. So all of these are gone because he cut that bond right there. Okay, we'll leave that bond just to show it. Okay, so he cut that bond with this hand. With the other hand, he does something special. He takes the 1 carbon. So again, this is the 1 carbon. This is the 6 carbon. He's going to make a bond between these two. This bond is, again, what? It's between the 1 carbon and the 6 carbon. What kind of bond would you call that, guys? You would call that bond a alpha-1,6 glycosidic bond. Okay, now look what that did for us. This glycogen synthase is like, you created some more surface area for me. I'm going to come over now, and I'm going to synthesize some more. So what he does is, he comes in, and he, again, what is he holding in one hand? In one hand, he's holding on to UDP linked to glucose. Here was our glucose, okay? So there was our glucose. And again, here's the 6 carbon group right there. In one hand, he's holding on to the glycogen molecule, right? He takes that UDP glucose and adds it on. And then what leaves is the process. So let's say I take and I add on 4 more of these guys. So what am I going to add on? 1, 2, 3, 4. Now that I added on these new 4, and again, what's going to be popping off to the side? 6 carbon, 6 carbon, 6 carbon, 6 carbon. I polymerized 4 more. When I polymerized 4 more of these, this was done by the enzyme glycogen synthase, because this is that enzyme. So this enzyme right here is glycogen synthase, and he's going to continue to keep elongating it. So again, what is this enzyme here doing? What is this enzyme doing? It's cutting this bond here between the 1 carbon and the actual 4 carbon. If it's cutting the bond between the 1 carbon and the 4 carbon, and it's an alpha-1,4 glycosidic bond, isn't that alpha-1,4 glucosidase activity? Yes, it is. That is one of the functions of this enzyme. So one function is you can break the alpha-1,4 glycosidic bond. So we call that alpha-1,4 glucosidase activity because it's breaking that bond. What was the other part of the enzyme doing? With the other hand, it was holding on to those glucose units that it cut, right? So it cut it like this point. So 1, 2, 3, 4, 5. It cut the alpha-1,4 glycosidic bond. And then it took these 1, 2, 3, 4, 5 glucose units and added it onto the 6 carbon of this glucose molecule and formed a alpha-1,6 glycosidic bond. So, in other words, it also has the ability to make alpha-1,6 glycosidic bonds. He has alpha-1,6 glycosidic bond activity. So he can make those bonds. Okay, what is this wonky enzyme then? This wonky enzyme here, he has a very complicated name. We're not going to use the complicated name. We're going to use the simpler name because that's what we want. We want simple, guys. This enzyme is called branching enzyme. Okay, so this enzyme right here is called the branching enzyme, and he has alpha-1,4 glucosidase and alpha-1,6 glycosidic bond activity. And if you can imagine, once he transfers that over, the glycogen synthase is going to go and polymerize alpha-1,4 glycosidic bonds. So now let's show the overall product. So I take this actual product we have here and we keep building on it to get the overall point now. So here's our glycogen molecule. And again, it's going to have that tyrosine amino acid, right? And then from that point on, you're going to have the glucose is being added. So let's say that we just do it like this now. So all of these bonds that I'm making at this point right now is alpha 1,4 glycosidic bonds. Who's doing this? Glycogen synthase. Then this enzyme, the branching enzyme, let's say he comes to right here, cuts this bond, takes all of this glucose units here from that 1,4 glycosidic bond that it cut and adds to the side over here. Let's say that that's the 6 carbon. Over here on the 6 carbon, it adds it right there and then does what? After it adds a couple of these units, who comes into play? Glycogen synthase. And then glycogen synthase actually makes more alpha 1,4 glycosidic bonds. So what was that guy doing? What was that branching enzyme doing? Cutting the alpha 1,4 and transferring those glucose units onto the 6 carbon to make alpha 1,6 glycosidic bonds. Once this is cut, who can come back into play? Glycogen synthase. And make what kind of bonds, guys? Alpha 1,4 glycosidic bonds. After he makes more of these, what enzyme will come into play? Let's say that this branching enzyme comes over here and cuts this bond. And then there's going to be a 6 carbon right here. What is he going to do? He's going to take these glucose units, transfer them onto that 6 carbon, and then do what? Then he's going to add these actual glucose units over here. And let's say that he adds these glucose units to about that point, and then who comes into play? Glycogen synthase, and he'll come and make more. And then glycogen synthase can elongate this. And it just keeps going and going and going. It's a nice interplay between these two enzymes, glycogen synthase and branching enzyme. What is this overall molecule now called? Glycogen. So we've synthesized our glycogen. We went through the process, guys. You guys made it through it, all right? So in the next video, now we're going to take this glycogen, we're going to break it down and show you how all of that activity is occurring. All right, Ninja Nerds, I hope this all made sense. I hope you guys enjoyed it. Until next time.